Okay, I think it's about time to get started. Good morning or good afternoon, depending on where you are, everyone. My name is Bryce Christensen. I am the Director of Services here at Golden Helix, and on behalf of our entire company, I would like to welcome you to our presentation today about knowing your NGS downstream functional predictions. We've had well in excess of 700 registrations for this particular event, so we're expecting a really good crowd today. And I hope you're all able to learn something new that maybe you didn't know before. Now, at any point during the presentation, if you happen to have questions, you can enter them in the question pane in the GoToWebinar software. And we will have some time at the end to answer as many of your questions as we can as we go along. Now, I would like to call your attention to one of our most recent webinars, which was given by Gabe Rudy, our Vice President of Product Development, about a month and a half ago. This was a very popular webinar that got a lot of good feedback. He talked about the vital importance of understanding how your sequence data was produced and how your analysis can be affected by some of the upstream protocols that are used. That webinar is recorded and available to view from our website at goldenhelix.com. And I'd like to point out that the feedback we received from that particular presentation really inspired what we're talking about today. We received a lot of requests for similar webinars about downstream analysis topics as well. So today's presentation, let's just outline some of the ground rules here before we go on. What I'm assuming about you is that you have some experience with sequencing technology and downstream analysis of genomic data, and that you're curious to learn more about the process and practice of functional predictions in sequence analysis. We'll talk about the informatics processes that underlie some of the more popular functional prediction algorithms, and we'll talk a little bit about how to apply these functional predictions in your own research. Unfortunately, we won't be revealing the true secret to making functional predictions. It's maybe just a little bit more complicated than that, unfortunately. So, you may have seen this slide or a similar slide many times in the past. There are different areas of sequencing analysis, and what we'll be discussing today falls directly in this realm of tertiary analysis, or the sense-making part of sequence analysis projects. And when you look at how sequence analysis is typically done in our field, there are several different workflows, but one of the really common workflows is based on annotation and filtering of variants to identify a set of plausible candidate variants that may be related to a particular disease or other phenotype. And so we see this workflow quite often of starting out with a VCF file, filtering out common variants, low quality variants, maybe filtering then by an inheritance pattern or a particular zygosity state if you're looking at a Mendelian disorder, then reducing to non-synonymous SNPs, and depending on what you've got left, it becomes very, very important to prioritize those candidate variants that remain. But the question of how to do that is really a major problem in the field. There are a lot of great methods for applying pathway analysis or for applying functional predictions, and that in particular is what we're going to talk about today. Now, if you look at the available algorithms for functional prediction, I've got a list of about 20 here, and this is not even a complete list. There are a lot of tools out there on the web if you go digging and looking for them. The starting of the inspiration for talking about this particular subject today, functional predictions, came earlier this year when I was sitting in a seminar about analysis of somatic variation in cancer, where the presenter rather casually mentioned that Polyphen II might be a little bit too biased toward germline analysis to be applicable to somatic variation. And when he said that, it really hit me quite hard that I didn't understand how these things worked well enough to really be able to apply them in my own work. And so that sent me on a path of discovery to learn more about all these various algorithms. And today I'd like to share with you some of what I have been learning as I've been reading the various papers and protocols about how to use functional predictions. Now, it seems on the top like maybe it's really a simple process. If you go look at all of the published comparisons that are out there that show how these various algorithms work, what you'll find is they mostly have really similar ROC curves, and you think, oh wow, it probably doesn't matter very much which one I use. They're all going to tell me what is damaging, what is not damaging in my data. But then if you actually run several of them, here I'm showing 5 popular algorithms, if you run all of these and then look at the actual intersection of what's predicted to be damaging and what's not predicted to be damaging, you find the agreement really isn't all that great. So this is from a particular sample that had about 10,000 total non-synonymous variants. Around 30% of those were predicted damaging by at least one of these 5 algorithms, but there's only 25 where they all agree it's damaging. So that's what we want to talk about today, is why they disagree with each other so much. So our agenda is going to go something like this. We'll start out by reviewing some of the underlying biology that is really the foundation for the functional predictions. A lot of us in sequence analysis, myself included, come from backgrounds either in mathematics or computer science, maybe haven't had a biology class for a while, so it's good to review some of these things. We'll proceed then with an overview of several commonly used algorithms. We'll look at the informatics principles that are driving the predictions from each one. We'll look at a few comparisons, see where they're similar, where they differ, and then proceed with a brief tutorial on how you can apply some of these predictions and what software is available for that. So let's go back to the beginning and review the central dogma of molecular biology. Francis Crick coined the phrase in 1958 that molecular biology deals with the detailed residue-by-residue transfer of sequential information. It states that such information cannot be transferred back from protein to either protein or nucleic acid. In other words, DNA is transcribed to RNA, RNA is translated to create proteins, and it's a one-way process. So that protein is where the damaging effects of DNA mutation will be observed. And quite often in DNA sequence analysis, particularly in discussing functional predictions, we refer to the DNA variant as being functionally important, but it's really important to recognize that it's the protein change that's expressed after translation that is really what is driving the phenotypic consequences. And all of the algorithms we're looking at today, for the most part, are basing their predictions solely on what's happening in the protein sequence. So quickly, just to review some terminology. Transcription is the process by which an RNA transcript is created from DNA within the cell nucleus. That messenger RNA transcript is then shipped out of the nucleus into the cytoplasm, where it can be then translated into protein. And the terms transcription and translation, I tend to get them mixed up a lot. You saw I just stumbled on them there a moment ago. But I think about it as translation converting to a different language. So nucleic acid as the primary language versus amino acid when we go from RNA into protein. And we see here in this little cartoon how each triplet of RNA bases is converted to a single protein, a single amino acid that forms the protein sequence. And we can see over here there's a standard code where each combination of 3 bases codes for a particular amino acid. And it's really important to talk about the different properties of each amino acid. So amino acids all have a common structural backbone, but then they also have this side chain, or R group, also called a residue, which is very different across the spectrum of amino acids. So we see a diagram here of the 20 different amino acids. Each one has particular chemical properties and particular formational properties that drive exactly what happens in terms of the higher level structure and function of the protein. So if we move on to the next slide, we can see that protein has several different levels of structure. So the primary structure being just the linear sequence of amino acids. And there are two standard structures that are then formed from that chain. Those are called alpha helices, or beta sheets, sometimes called beta slides. And those are regular structures that appear in almost all proteins. And those structures then interact with each other to form a more complex tertiary structure. The tertiary structure is influenced by attractions between the various secondary structures. And then proteins also commonly have a quaternary structure, where there are several different amino acid chains, or several different tertiary structures that all interact with each other to form a superstructure here. Now, the structure of each protein largely determines its function. Proteins include various types of functional domains, binding sites, and other surface features. Those determine how the protein interacts with other molecules in a cell. And if we think back about how all of those various various amino acids have different charge, different polarity, different shape and size, if you replace any given amino acid in a protein, it may or may not have really drastic effects on the protein structure, thereby affecting the protein function. And if we know how the protein structure is affected by any given amino acid substitution, we can then make a pretty good guess about the functional consequences of that mutation. But the big problem is that we don't know the wild-type 3D structure of most proteins. I remember probably 10, 12 years ago now, sitting in my first ever bioinformatics seminar and hearing the speaker discuss prediction of tertiary structure as being the holy grail of bioinformatics. And I don't think I fully appreciated what he meant by that until just recently. I'm starting to realize that, wow, if we could accurately predict the tertiary structures of all of these proteins, we could make significantly better predictions. As it is, though, we have to use what information we do have as a proxy for the tertiary structure, and therefore a proxy for determining what is really going to be functional in consequence. So if we go back a few years to 2001, there's an important paper that found that 83% of disease-causing mutations known at the time affected the stability of a protein, and that 90% of disease-causing mutations could be detected using structure and stability criteria. Now, we know that most, if not all, human proteins have numerous homologs. There's two different types of homologs. So a homolog is a second or third, just an additional protein that has a very similar primary sequence or a very similar structure. And it could either be a paralog, which is separated by a gene duplication event, so a second human protein that at some point in time has been copied and appears either on a different chromosome or elsewhere on the same chromosome. And then there are orthologs, which are homologs separated by speciation events. So if we look in evolutionary relatives of humans, we can find the very same protein very often, and it's the result of having a common evolutionary ancestor. Now, we don't know the exact structure of most proteins, as we've discussed, but we can usually compare the amino acid sequences to identify the various sequence domains and motifs that have been conserved by evolution. And as this other paper from about the same time period found, that the disease-causing mutations are overrepresented at conserved sites in the primary structure. So when we look at all of these homologous proteins, we can usually figure out what areas are going to be important for phenotypic and or disease consequences. And we do this by utilizing what's called a multiple sequence alignment, or MSA. And we'll be talking a lot about MSAs as we go along today. But you can generate an MSA using BLAST or a few other similar algorithms that exist. BLAST is by far the most common. And all of the functional prediction algorithms we'll look at today incorporate MSAs, and almost all of the contemporary algorithms that are out there do the same. And so what we're looking at here in this diagram is 3 different protein families, the P53, P63, and P73, that have high homology to each other. And you can see in the vertical columns how each amino acid is, in many cases, very similar, highly conserved. So we see this R, which I believe is arginine as the amino acid, is conserved throughout all of the proteins, all of the different homologs represented. While at other positions, we see less conservation. So here with the QH, P, Q, several different possibilities there. And as we look at the different algorithms today, you'll see that they all take advantage of this type of a process. And the MSAs may include 700 or more homologous sequences. Some of them get very large. And the prediction algorithms, they could incorporate orthologs or paralogs in the MSA, or both. The distantly related orthologs, and remember orthologs are the ones separated by speciation, they're frequently cited, especially from the SIFT authors I've found, as giving the optimum prediction performance. But there are also warnings that I've seen in the literature that the phylogenetic relationship doesn't always mean that the protein has the same function or is similarly important in both species. And some authors really argue you need to use both paralogs and orthologs. And while most algorithms incorporate MSA, what they really differ in is how it's interpreted and scored in the process. So there are 2 main classifications of prediction algorithms that we'll look at today. There are trained or weighted algorithms, and there are untrained algorithms. The trained algorithms use the machine learning method and classify the functional consequence of any given mutation based on characteristics observed in a canonical set of mutations that are known to be damaging or benign. And there are different ways that they might weight the results. For example, they might include known disease sequence in the sequence alignment and so on. But the important thing to recognize here is that the selection of training data is really an important factor in the algorithm performance and the appropriateness of using that algorithm in your analysis. Now, the second option, the untrained algorithms, of course, don't use a machine learning process, but they're based on a theoretical model, usually incorporates a lot of important prior knowledge about what to expect in disease-causing mutations, but they don't necessarily have the same biases that might be present in some trained algorithms. So with that as a background, let's go ahead and review a few specific algorithms and see a little bit more how exactly they work. We're going to look at 5 algorithms today: SIFT, PolyPhen-2, Mutation Assessor, Mutation Taster, and FATHM. Now, the reason these 5 were selected is because they're all included in a really useful tool called the Database for Non-Synonymous Functional Predictions, or DBNSFP. And that's a tool that can be accessed within our Golden Helix SVS software package. As we go along, you'll notice that on several slides there are statements that are in quotation marks and highlighted in blue. When you see that, that represents a statement that was taken directly from the algorithm's own website or from the original paper describing that particular method. Now also you'll see that I have citations for when it was published and how frequently it's been cited in the literature. Be aware that usually we're looking at the paper that describes the current public version of the algorithm, and quite often there are earlier papers that describe either previous versions or that describe the underlying foundational science behind the method. Now, I already mentioned DBNSFP. It's a free tool developed by Xiaoming Liu at the University of Texas. It catalogs pre-computed conservation and functional prediction scores for all possible non-synonymous SNPs in the genome. I think the number is around 75 million, if I remember correctly. It's downloadable. It comes with a Java program that you can just feed a VCF file directly into it and get a lot of rich information back, including conservation scores, functional predictions, and a lot of other useful annotations. And so we used DBNSFP for generating some of the data that you'll see in the algorithm comparisons today. So the first one we'll talk about is the SIFT algorithm, or sorting intolerant from tolerant. SIFT is a very popular algorithm. It's been around for a long time. It hasn't changed a whole lot over the years. You do see there are several different papers, earliest one here describing the original method, then describing the publicly available version, as well as a follow-up paper in Nature Protocols just a few years ago that describes the best practices for using it. Now, if we look at how it really works, SIFT relies completely on protein sequence and does not include any structural features of the protein. It builds a sequence alignment based on BLAST and considers a whole bunch of different features in scoring a variant amino acid. So it asks questions like this: is the position highly conserved for a single amino acid? So you can see in this column in the example, always having the L. That's a highly conserved position. Is it highly conserved for amino acids with a particular polarity, charge, or other chemical property? We see over here on the left a column where all of the possibilities present in the alignment are hydrophobic in nature. And then it asks, how different is the mutant allele from the most common allele? So even if it's not a highly conserved position, it will look and see what's the most common, what have I actually created with my non-synonymous SNP, and are they chemically interchangeable or are they very different? And so the product of SIFT then is a set of predictions where each possible substitution is predicted either as tolerated or not tolerated. And I believe these two figures refer to the same data, and you can see down here at this last position, H, the histidine residue, is what was originally observed and the only one that would be tolerated at that position. Anything else would be considered damaging. So we see in this matrix of scores below the H, that's the only one that's scored as tolerated. Anything in red would be considered intolerated. The cutoff score is usually 0.05. Now, one of the caveats with SIFT that is clearly explained in the various papers is this concept of sequence diversity versus prediction accuracy. And they say that confidence in a substitution predicted to be deleterious depends on the diversity of sequences in the alignment. And if the sequences used for prediction are closely related, then many positions will wrongly appear to be conserved, leading to a high false positive error rate. And if they give this nice graphical illustration of that, you can see here in black the prediction accuracy for all variants compared to the conservation across the sequences used in the alignment. And you can see that up here above 3, the overall accuracy starts to drop. The true positive prediction goes up, but we also have a very big problem with false positives starting to appear above that. So they recommend that predictions with a median conservation score above 3.25 should not be used. And this is a problem that I think may affect other algorithms as well. They all have different ways of dealing with it, but it's important to realize that if a protein sequence is really unique and doesn't have very many homologs available, it's going to be difficult to accurately predict any mutations or the effect of any mutations. So SIFT has a nice web interface available for making queries, be run interactively or through a batch upload, and it does provide extensive annotations with the output. So really encourage you to take a look at it. One of the special things is that it gives classification information for indels. It doesn't give a tolerance score, but it does give some other useful information, like whether it might lead to nonsense-mediated decay and so on. Quite often when I talk to people just getting started in sequence analysis, one of their big questions is, what do I do with indels? And SIFT is a good place to start that can give you some ideas about which ones might have functional consequence. So moving on, let's talk a little bit about Polyphen II, or polymorphism phenotyping version 2 as it's called. The current implementation was described in 2010. The original Polyphen goes back about a decade before that, although Polyphen II is substantially different than the original method and can't really be compared directly. So let's look at how it works. So Polyphen II is a trained algorithm that uses naive Bayes classifiers to score variance based on 11 predictive features. And the features that they claim or state that are most important are this idea of how likely the two alleles, so the wild type and the alternate, how likely they are to occupy the site given the pattern of replacements seen in the MSA. And this is based on the PSIC score, which has been around for several years and is based on a Blossom substitution matrix if you really want to know the details. Then they also look at how distant the protein harboring the first deviation from the human wild type allele is from human protein. So if you can think about taking all of the sequences that appear in the alignment and sorting them by just how different they are, how distantly related they are in an evolutionary sense, we look to see how far down the chain you have to go before you find any sort of mutation. And then it also considers whether the mutant allele is at a hypermutable position. One thing I'll point out in this little diagram is this concept that when it builds the MSA, it limits to a maximum identity of 94%, which I think is how they're adjusting for that concept we saw with SIFT, where if the sequences are too similar, it can negatively affect prediction results. So they're adjusting for it when they're creating the alignment rather than scoring the effect later on. So if we look at the actual prediction model, as we mentioned, there are 11 features that are included, 8 of them based on sequence, 3 based on structural features for the proteins that actually have a known structure. And they do state that they think orthologs and paralogs should both be included in the MSA, perhaps because a majority of disease-causing replacements affect protein structure rather than specific aspects of function. And so they're saying that both orthologs and paralogs should be structurally similar, even though they might be functionally different than the original protein. And it's interesting to point out that in this statement, inherently, you can see that they are really interested in identifying disease-causing mutations rather than anything that simply has some phenotypic effect. You see throughout the literature that it's the variants that cause a structural change are the ones that really are most likely to lead to disease. So we won't go through all of the different features, but the lists are here, and you can see that they cover a pretty wide range of topics. Now, the most important thing to understand about Polyphen II, in my opinion, is that there are 2 different prediction models available within the algorithm. And one of them is called HUMVAR. It's a human variation-based panel that they use for the training data set, and that's where they differ from each other, is in the training data used. But it's trained on disease-causing mutations from the UniProt database together with a variety of human non-synonymous SNPs that are not annotated to be involved in disease. But it's expected that those non-damaging SNPs still include a sizable fraction of SNPs with mild phenotypic effects. And the idea is that that particular prediction is really tuned to detect drastic effects and is best used in analysis of Mendelian traits. Now, on the other hand, if you are analyzing a complex disease, they recommend using the HumDiv panel, or the human diversity is what I believe it is representing. And so this one is comparing known damaging human alleles against a set of 6,321 differences that have been observed between human proteins and closely related mammalian homologs that are assumed to be non-damaging and selectively neutral. And therefore, it's expected that it will detect even mildly deleterious alleles as being damaging and is better to use when you're looking at a complex multigenic disease. Now, just like SIFT, PolyPhen-2 has a nice web interface. It includes options for both an interactive or a batch query system. They also have a standalone version that you can download and install locally. One thing I would say to watch out for is the documentation and user guides are not complete. The website says they are under construction and have been for quite some time. And then also point out this classifier model down here for HumDiv or HumVar. It's listed under advanced options, but it's really important to consider that and make sure you don't skip over it if you decide to use PolyPhen II. So with that, let's move on and talk about the next one in line, Mutation Assessor. This is a much newer algorithm. First, well, the current public implementation was described about 2 years ago. But it's one that I've heard a lot of positive things about recently, and I myself have come to really like it. So if we look at what it does, Mutation Assessor is unique in that it was designed with special consideration for evaluating somatic variation in cancer, although they frequently remind us that it's also useful for germline analysis, and I agree with that. But the authors are careful in their selection of terminology. They refer to everything as being functional rather than damaging or disease-causing. And their concept is to try to capture different types of variation. So loss of function, gain of function, drug resistance variance, and what they refer to as switch of function variation, all of which can be important in cancer, but also in other diseases as well. They have a nice figure in their paper that looks at the functional impact score that is produced by Mutation Assessor and how it increases both in known loss of function and gain of function variance in the TP53 transcriptional path. So if we look at how it works and what it does different than the others, it uses multiple sequence alignments together with 3D structures to some extent, although it seems that the 3D structures are really just annotated in the output rather than being calculated as part of the impact score. But it stands out from others in the way it uses protein subfamilies. And it calculates 2 scores for each amino acid. There's the overall conservation score. You see an example here of a conserved amino acid, which is consistently a G, I believe that's glycine, throughout the entire protein family. Then they calculate what they call a specificity score, and I haven't seen this in any of the others, which indicates that it's conserved within the subfamily, but not necessarily across the entire protein family and the entire sequence alignment. And the final impact score is the sum of those 2 scores, conservation and specificity. And going back to the specificity, they did some additional analysis on that and point out that those residues are predominantly located on protein surfaces in known or predicted binding interfaces, often directly linked to protein functional interactions. So that idea of looking at subfamilies and subfamily-specific conservation is really what's driving Mutation Assessor as being different from the others. They have some nice schematics in the paper that show how, if you look at non-disease polymorphisms versus disease-causing, that their score is always shifted higher for disease association. But specifically for cancer, when they looked at the COSMIC database and looked at somatic variants that are observed frequently, highly recurrent, that those types of variants consistently have very high functional impact scores with Mutation Assessor, while those observed not quite as often or not as high, and those observed only once across all of the thousands of samples in COSMIC are again shifted just a little bit lower. So it seems to capture the effect of the most significant mutations quite well. And here's just another schematic that I think is important not only for interpreting Mutation Assessor but also some of the others, which is that they can draw this curve indicating what proportion of all damaging variants are scored above a particular threshold. And we can see that if they draw the threshold in at about 1.9 on their impact score, 78% of disease-associated variants score above that threshold. A similar proportion of non-disease variants score below. And so there is some crossover between the groups. The predictions are not always going to be clean. They just have to pick an optimal threshold and go with it. And so you can see that it scores variants as neutral, low, medium, or high. And the medium and high group are the ones that are generally predicted functional. Now the Mutation Assessor website is really nice. I've had a lot of fun working with it. It gives extensive output and it has some really nice links to external data sources. I'd like to just take a quick look at it here with you, if you don't mind. So if I open up my web browser and go to the MutationAssessor.org website, what I'll do here is just run the set of example variants that it allows you to input. You can also enter specific coding changes based on DNA coordinates. Here we're just looking at a variety of different amino acid substitutions in EGFR. If I submit this and let it run, and I'm just using default settings, there are a lot of additional options I could select. It goes through all of the mutations in that list and scores them all as expected as neutral, low, medium, or high. It tells me the functional impact score. Tells me that all of these had a very deep sequence alignment that the predictions are based on. I think 700 is the maximum where it stops. It also tells me which ones are in functional regions, if they're in protein binding sites. There are links to other sources that will tell me more about any small molecule binding sites going on. But what's really fun and really valuable, I believe, is that it also gives a link to show us the actual sequence alignment used in the prediction. So we can look at the highlighted position. This is the variant that we selected and see up and down what the conservation is. So we see that it has a relatively high conservation score, kind of a moderate specificity score. And we can also find out a little bit about the related sequences. And then it also gives me a link to a 3D model of the protein from Protein Data Bank. Now in this case where it says identity 100%, that indicates that we are looking at probably the very same protein. I've seen some examples where the most similar protein that it could draw a rendering of was only 40 or 50% similar. But when we get this 3D model, now I can rotate it around, maybe zoom in, see exactly where my mutation is, see where it is relative to highly conserved or highly specific variants. And if you get really good at using this, you'll find that there are a lot of different options for adjusting the view and being able to highlight specific functional domains and get an idea of where your particular variant lies. And with my own background being more in statistics rather than biology, I may not be able to interpret this very well, but I find that a lot of biologists really enjoy this sort of an analysis. So let's come back now to our slides and move on. The next one we'll look at is Mutation Taster, and we'll just quickly review Mutation Taster. It's produced by a group in Berlin, Germany, and it's a trained classifier in many ways similar to PolyPhen, but the thing that really stands out about Mutation Taster is that it has native support for DNA alterations rather than amino acid substitutions, and because of that, it is able to give different types of annotation that we don't see elsewhere. For example, it annotates indels and non-coding regions of the genome in addition to the protein coding SNPs. And another interesting feature is that it can combine adjacent mutations into a more complex substitution polymorphism to figure out what is the real amino acid change. So you might find in your data that you have places where there are consecutive bases called with SNPs. They may or may not be on the same haplotype, but if they are, it's possible that each SNP individually might produce a different amino acid, while combining them would produce yet a third amino acid. And so it's important to recognize that that is a possibility here. And Mutation Taster tries to adjust for that. And perhaps my favorite thing about Mutation Taster is that it has a web interface that allows you to directly upload a VCF file. And that seems to be a popular feature. When I looked at it a few weeks ago, there were over 6,000 VCF files queued up for analysis. When I looked yesterday, it was down less than 1,000. It seems to be tapering off a little bit. But that is a very useful feature because we all have VCF files, and all of the other algorithms require some informatics work to translate it into the correct input format. So we won't go into too much more detail about Mutation Taster, except to point out that it does use 3 different classification systems depending on whether your mutation is in a coding or a non-coding region, and if it's coding, if it's affecting a single amino acid or several. And in the non-coding regions, there's not a lot that we can tell in those regions, but what they do provide is information about alterations of Kozak consensus sequence, which is a motif that generally appears near the transcription start site or the binding sites. And then it also gives some information about the propensity of a mutation to affect splicing. And this is kind of a tricky one I've found. When I've looked at their output, it seems like just about every SNP in there has some ability to affect splice sites. So be aware of that as you're looking at it. Some other nice annotations from different external databases and conservation scores. It does have some quirks, and I won't go into too much detail, but for example, I've seen that the web interface gives a few extra variables that you won't get from the batch system with VCF files. I ran a few example VCFs through where it seemed like output may have been truncated in some of my results. And one of the really interesting ones, I noticed that I had a SNP in a gene with 4 different transcripts, but the SNP was actually way like 600 bases downstream of the 3' end of a few transcripts, but it was still annotated as being in those transcripts and affecting splicing of them. And so just some little quirks like that, but once you get used to it and figure out the pattern there, you'll find that Mutation Taster is very helpful. And so the last one we'll talk about here is Fathom. Fathom is very new. It comes from the University of Bristol in the UK. And it's new enough that it still hasn't really started to generate any citations, so it's hard to tell how much it's getting used. But the thing that really separates it from the others is that they use a different technology for building the sequence alignment. So they make the argument that sequence alignments based on BLAST have some inherent flaws, and that a model based on hidden Markov models is much more efficient or more correct, I guess you would say, than the BLAST method without going into too many details there. So the standard untrained version of Fathom uses these hidden Markov models to make the MSA, and then that is used to assess conservation of amino acids. In a lot of ways it seems similar to SIFT. In fact, they cite the original SIFT paper as being one of their primary inspirations, if you will, for creating Fathom. But then it also takes an extra step of looking at a manually curated library of alignments for conserved protein domain families taken from the PFAM and superfamily resources. And they have this additional extension, a species-specific version or human-specific version, if you will, that incorporates pathogenicity weights. And really what's going on there is they check the MSA alignment to look for any known disease-associated sequences and look at the relative frequency of those disease sequences versus known neutral sequences and weight the results accordingly. And that works out, as we can see here on the left, is a diagram of the effect scores on the unweighted method, and the darker grouping here is the known damaging set of mutations. They generally have lower scores, but there's not a really clean separation between the two groups. While over to the right, this is based on the weighted method. Here the separation is quite a bit cleaner, and they're able to get a very nice prediction on the functional versus non-functional mutations. And if you want to use Fathom, they also, like the others, have their own web portal. You could submit variants based on amino acid or by RSID. They don't support any other DNA-based formats as yet. They have a downloadable application that you can run locally. And interestingly, they have a cancer-specific version available on the website. It's not yet published. I think that it uses the same weighting method, but specifically weighted to identify known cancer-related variants. I'm not positive about that, but that seems to be what's going on. The paper describing the cancer version is in press, it says. And so with that, we've now reviewed 5 different algorithms. We can kind of see that there's some core similarities in the underlying informatics, but there's also some real differences in how they calculate functional scores. And the natural question is, how do we know if these things really work, and what should we use in our actual research? And if you look around on PubMed, you'll find a lot of published comparisons based on various standards that look at the performance of the various algorithms. And those comparisons are a good starting point to really understanding what makes these differences in the methods. And here we see a few figures from a paper that I found very helpful. It's a 2010 paper by Thusberg and others that shows over here on the left this accuracy metric for several different methods. And we can see most of them have similar accuracy for the particular benchmarking data that they used. Most of them are between 70 and 80% accurate, with some notable exceptions. And similarly, we see the sensitivity scores where there's a little bit more variation. If we really dig into it and look at some of the different comparisons that are out there, you see a pattern emerge of accuracy rates overall generally being in the 65 to 80% range. So this is from the Fathom paper, where they took the comparison method from the Thusberg paper and added in their own method to show how it would compare to the others. And here we see, again, accuracy scores between 65% up to 80, 86% in one case. And I don't have any ROC curves here to show, but you generally see they've got pretty good sensitivity and specificity. Issue is that in practice, there's a lot of variance with unknown consequences, and that makes interpretation really difficult. If you break it down, you'll see most of these algorithms will predict between 10 and 20% of all non-synonymous SNPs to be damaging. And here I've taken 2 samples and taken all of the non-synonymous SNPs in those samples based on sequencing done by Complete Genomics and classified them with each of the 5 methods we've described. And there's a few patterns that really come out when we do this. The first pattern you'll see is that Fathom is very different from the others. it calls a total of 1,000 out of just over 10,000 variants to be damaging. But 3/4 roughly of the variants it calls damaging are not considered damaging by any of the other algorithms. And we see over here in the second sample, a Puerto Rican sample, the same pattern of a very high proportion not having any crossover. The second kind of consistent pattern I've seen is that when we look at the intersections, the SIFT and PolyPhen intersection is usually pretty high, as is the three-way intersection here of SIFT, PolyPhen, and Mutation Assessor. Those three generally tend to be quite similar. Also, we see PolyPhen and Mutation Assessor being a pretty large intersection group, as well as down here SIFT and Mutation Assessor. So those three all seem to go together. And lastly, Mutation Taster always tends to be the most conservative. It always has the fewest number of mutations that it considers to be damaging. We can look at two more samples from different parts of the world. We see really identical patterns going on over here. If we build a table then of how many of these 5 algorithms call any given non-synonymous SNP to be damaging or functional, we can see that in all 4 of the samples we looked at, right around 10,000 non-synonymous SNPs, and a very small number of them are consistently called damaging, while there's really some wild differences in each individual algorithm. Now, of the 23 SNPs here in this cell, so this is in a European sample, NA12878, which is a very well characterized sample that's used in a lot of benchmarking processes, we can see that of those 23 that are uniformly called damaging, 13 of them are in 1,000 Genomes Project, and 11 have allele frequencies greater than 1% there. Of course, they should all be in 1,000 genomes because that sample is there, but this is based on the complete genomic sequence, which may call some different variants than 1,000 genomes had. But then, additionally, 15 of those 23 are in the NHLBI exome sequencing project data, and 8 of them are quite common there. So even when we get to these really apparently damaging variants, we'll find that some of them are still pretty common. It may just be that they have some basic phenotypic consequences that aren't necessarily going to be disease-causing or really seriously damaging. And the last thing I'll call your attention to here is that this one African sample has maybe 15% more total SNPs called than the others, but disproportionate number that are considered damaging by all 5 methods. And we know that African genomes are very diverse. It's well understood that the human reference genome that these are all being compared to is biased toward European alleles. But it looks like the protein sequences used in the sequence alignments are similarly biased, and I think that's why we're seeing kind of a disproportionate number of damaging variants being identified in Africans. So that's something to watch out for if you are working with African data. Now, when it comes down to answering the question, which one should I use, the common belief that a lot of people seem to be following is that if a variant is called damaging by multiple algorithms, it's most likely to be the true disease-causing variant. But remember, the published comparisons that are out there aren't exhaustive. They usually focus on performance of the algorithms for a particular category of mutations, and each tool has its own strengths and weaknesses. Really, it just takes some time working with them to get a feel for how they work and learn which ones you like and which ones you don't like. And as we can kind of see in those Venn diagrams we were looking at, that the false positive rate is likely to be high when you apply these methods to a broad range of variants of unknown significance. If you can reduce your variance based on other factors, perhaps based on genes that are in a pathway expected to be related to the phenotype you're studying, it can really help with improving the prediction rates. I can just say from anecdotal experience, a few things that I have noticed in trying to compare these, that the algorithms individually consider a lot of different factors, and if you pick out a variant that's damaging in one and not in the other, it's really hard to identify an obvious reason for the discrepancies. I've seen, by looking at things that are neutral in Mutation Assessor but damaging in SIFT and PolyPhen, quite often the alignment depth in the MSA is really low, and so it's just a lack of available data to use for Mutation Assessor. Similarly, if you look at what's called by Polyphen and Mutation Assessor but not SIFT, the thing that stands out to me, and maybe it's just because that's what I was looking for, is that the reference and alternate amino acids usually looked like they had similar chemical properties, and maybe SIFT is more sensitive to that than the others. But it's really hard to identify a smoking gun. So finally, what I'd like to talk about is how you can apply functional predictions in your own work. And we are running a little low on time here, but I'll just quickly mention that if you have access to the Golden Helix SNP and Variation Suite software, we do have the DBNSFP that we've talked about already today integrated in the software. You can use it to annotate and filter your SNPs based on functional predictions from DBNSFP. You can use any or all of the 5 algorithms we've talked about today. You can also view that prediction data interactively in our Genome Browse product. And I won't take the time right now to demonstrate the actual nuts and bolts of how it works, but it is there, it's easily available. Also, DBNSFP, as I've mentioned, does have a downloadable Java program that you can use directly on your VCF files if you would like. And I think we'll skip the demo for today just because of time constraints, but if you have any particular questions about how SVS works, you can always call us and ask, or check out our website. There is a tutorial there that specifically describes how the DBNSFP filtering process works. And in closing, I'd just like to mention a couple papers that I have found very helpful in preparing this presentation. You saw throughout I had a few citations to various sources. I want to highlight these. This 2006 paper, it's actually from the authors of the SIFT method, but it's a nice review of the science behind functional predictions. And also this paper from 2 years ago that is a very thorough comparison of all of the methods and their performance in predicting the effect of missense variation. So, as we mentioned earlier today at the beginning of the presentation, this topic was the result of feedback that we received from you, from the community, and you will all be receiving an email. Everyone who's attending the presentation today will be receiving an email later asking for your feedback. And if there are any particular topics that you would like to know more about, let us know, and we may give a presentation about your selected topic. We are doing this for you. And with that, I'd like to thank you all for your attendance today and ask if you have any questions. So I know that we are right at the end of the originally scheduled time, so if anybody needs to leave, don't feel bad about stepping away. But we will go ahead now and try to answer as many of these questions as we can. All right, so looking at the questions, there's one from Daniel asking, I'd like to know how you generated the Venn-style diagram in the different methods. That is actually a plot that we can generate inside of the SVS software using a tool called Compare Variance Across Several Spreadsheets, I believe is what it's called. It's based on a Python plotting library. Another question from Tammy. Is there any way to predict whether a homolog is a functional ortholog when comparing between different model organisms? It seems that based on pathways or other protein-protein interaction information, functional orthologs could be predicted. You know, I'm sure that there probably is a method for doing that. I know in particular some of the methods we've looked at today do give you some control over which sequences you use for building your sequence alignment. There's probably some way to adjust what you're including there to get an answer to your specific question. Honestly, that's a little bit beyond my area of expertise, but it's a very interesting question. Comment from Christian, which I appreciate. BLAST cannot construct MSAs, only pairwise alignments, and that's really just a, I'll call it a semantic difference, maybe something that I didn't properly understand, just used some wrong language there. Another question, are all of these algorithms intended for human genes? Is there any that would be appropriate for plant genes? So a few of them are definitely applicable to additional species beyond human, and there's a lot of low-level user control for controlling which databases you use for constructing your alignments. Particularly with SIFT, you have that ability. And some of the algorithms that I did not demonstrate today, I'm trying to remember particularly, I think Panther in particular can help with that. It's a very good question. And finally, is there any good algorithm to predict functional consequence of an indel, including frame shift and in-frame shift mutation? As far as the things that we've looked at today, the one that I like best for doing that is probably Mutation Taster. If you look at the output it gives for indels, it does look at the entire length of the protein. It identifies whether it's going to introduce an early stop codon later on. It calculates the probability of nonsense-mediated decay. And I've found that one to be useful and probably about the best one there. Also, SIFT has some helpful functionality there. So there are a few other questions here. A lot of them are similar to what we've already answered. One thing that is coming up here frequently is people would still like to maybe see that demo that we skipped over. So I'll just take about 5 minutes now to walk through a brief demonstration of how this works in the software, and we'll go ahead and close with that. So we come back over here to the screen and jump out and open SVS. So if you're not familiar with it, this is the Golden Helix SNP and Variation Suite software. And I have a project here where I'm looking at this same European sample that we've talked about a few times already in the presentation, looking at the exome sequence. It's already gone through a series of quality filters, and I've subset the data to non-synonymous SNPs that are rare in the exome sequencing project. So I can look now at that particular spreadsheet and see that there are about 3,000 variants that survived through those filters. And if I want to apply functional predictions to this, I can come to my Select menu, and I have a tool here called Filter on Non-Synonymous Functional Predictions. And this brings up a menu with a lot of options for selecting exactly what it is we want to look for based on DBNSFP. But we'll go ahead and run it with the default settings, which is set up to drop out any variant that's uniformly predicted to be benign or well tolerated. So we'll go ahead and run that. And we see that up here in the original spreadsheet, a number of columns have been turned gray or inactivated. And there's the message here that tells us 903 non-synonymous variants in the data met filter criteria and were therefore dropped out. Then also, looking at the report that's generated, we can see a lot of information about each variant in the original spreadsheet, including SIFT scores, Polyphen scores, Mutation Taster, Mutation Assessor, and Fathom, as well as a few additional conservation scores, CHIRP++ and PhyloP. Now, I could at this point continue working in the variant spreadsheet that's had the tolerated variants removed. But suppose I wanted to follow a different path and perhaps pick out a particular pattern here. So if what I really wanted was to consider only the predictions from SIFT, PolyPhen, and Mutation Assessor, The software allows me to come in and specify the pattern I'm looking for. Now suppose I wanted to find where SIFT is damaging, where Polyphen II is also damaging, while Mutation Assessor says it's strictly neutral. I can run that. It takes just a minute to finish. It gives me a report on how many categories each variant passed. Then also I see only 12 of my remaining variants fit that category. And I'll just join this with some of my other classification data here to generate sort of a master list of what I know about these 12 variants that are neutral according to Mutation Assessor, but damaging according to the other 2. So I can scroll through, see all of my information. I can see what gene they're in, what's the actual coding change and protein change there. One thing that's interesting is to go back and look at these and find out from the original source maybe why Mutation Assessor in particular called them to be neutral. And I already have that up and running here in my web browser. So I have those very same 12 mutations already ready to go. If I submit them to Mutation Assessor, the thing that really jumps out to me is that of those 12, over half of them have very low sequence alignment height. And so those are things where Mutation Assessor probably didn't have enough information to make a really good estimate, and it seems to default to a neutral impact in those cases. We can also see some had really good depth of coverage or in the alignment. And we see one here also is in a functional region, a known protein binding site, a small molecule binding site. So maybe that one is interesting to look at. So we can pull up the sequence alignment there and see right away it is a location that has a high specificity score, so it's conserved within protein subfamilies, although not conserved overall so highly, which is interesting because SIFT and PolyPhen, which called it damaging, rely heavily on overall conservation. So this one is a bit of an enigma as to why it's not damaging in Mutation Assessor. It seems like maybe it should be the other way around. And if we look at some of these same variants in SIFT, I've also pre-run them here, and we see that SIFT actually has a higher number of sequences at each position. So it's clearly using a different alignment tool or a different tool to generate the MSA, or at least with some different rules. And this is what I was kind of getting at earlier when I mentioned it's hard to pick out the smoking gun. There's not a lot of information presented about the internal workings of the algorithm, but it considers so many different variables with so many different interactions that it's just really challenging to finally know the answer. But you can see here how we are able to apply predictions from DBNSFP, get a prioritized or at least a subset list of variants that fit the particular pattern we're interested in, and we can take those out and just assess their consequences using other databases as well. So with that, I think I will go ahead and wrap this up. I see we've got a lot more questions that have come in. We'll try to reply to as many of those as we can by email later on. And again, I thank you all for your attendance. And let us know what you think and if you'd like to see any more additional topics later on. Thank you and have a good day.